Welcome to your personalized deep dive. You're interested in sarcoma research.
Oh, absolutely.
So we're going to take you on a journey into this really complex field.
Yeah, sarcomas are kind of like the rebels of the cancer world.
Okay.
They're this rare and diverse group of cancers that can pop up in various tissues,
Wow.
and they affect both children and adults.
Yeah, I was reading that there were over 50 types of sarcoma.
Right.
Right.
Yeah.
That must make them really hard to diagnose and treat.
Absolutely. It makes them super challenging to both diagnose and treat and research.
Yeah.
And that's really where this new tool that we're going to be exploring comes in.
Okay.
It's called Sarcoma CellMinerCDB, and it's essentially a massive database
Right.
packed with information on tons of different sarcoma cell lines.
Okay. I'm intrigued. The article that you sent me from iScience describes it as a one-stop shop for sarcoma researchers.
Yeah.
What kind of information are we talking about?
So think of it as like a detailed profile for each cell line. We're talking about their genes, how active those genes are, how they respond to different drugs,
Wow.
and even how they control their own DNA.
Whoa. That's a lot of data, but
Yeah.
how do we know if it's reliable? I mean, garbage in, garbage out, right?
You're totally right to be cautious about the data reliability. It was key for these researchers.
Yeah.
So they took great pains to compare data from different sources,
Okay.
and they actually found a high degree of consistency.
Oh, that's good to hear. Do you have an example?
Yeah, so take the SLFN11 gene.
Okay.
Its expression levels were remarkably similar across all these different databases.
Okay.
And this kind of consistency was seen for other types of data, too, like methylation and copy number.
Okay, so it sounds like we can trust the data in this Sarcoma CellMiner CDB thing.
Yes, you can.
But how does it actually help researchers?
Well, one of the biggest advantages is that it lets researchers zoom in on specific subtypes of sarcoma.
Okay.
Remember, these cancers are incredibly diverse.
So instead of looking at sarcomas as like this big blob, they can study them in a more targeted way.
Precisely.
Hmm.
For instance, let's look at MDM2. This is a gene that's often amplified in a type of liposarcoma. Sarcoma CellMiner-CDB shows that this gene has the highest expression levels in the cell lines where we would expect it to be based on what we know about liposarcoma.
Ah, so it's like the database confirms what we already know about how sarcomas behave in real life.
Exactly.
Okay.
And it goes even further. Sarcoma CellMiner-CDB is fantastic for studying gene fusions.
Now, to be honest, I need a quick refresher on gene fusions.
Okay, so imagine two songs
Okay.
getting mashed up
Okay.
to create this brand new track.
Uh-huh.
Sometimes that happens with genes, too.
Okay.
Pieces of two separate genes get fused together,
Okay.
creating this hybrid gene that can actually drive cancer growth.
That makes sense. So does this tool help identify those cancer-causing mashups?
Yes, it does.
Okay.
Take Ewing sarcoma, for example.
Okay.
The database confirms that the EWS-FLI1 fusion is present in almost every Ewing sarcoma cell line.
Wow.
There's only two exceptions.
Okay.
And they have a different fusion called EWSERG.
Okay.
And guess what?
What?
That's exactly what we see in patients with Ewing sarcoma.
So it's like this database is mirroring what we already know about sarcomas, but in a much more detailed and organized way.
Exactly. And it's not just confirming what we know.
Okay.
It can also help us uncover new information about mutations,
Okay.
including something called tumor mutational burden,
Okay.
or TMB.
Okay. I have a feeling I'm going to need another quick explainer here.
Sure.
What exactly is TMB?
Think of it like a measure of how many typos are in a cell's DNA.
Okay.
Some cancers have a lot of typos,
Uh-huh.
which means they have a high TMB.
Okay.
Others have very few, indicating a low TMB.
Interesting. So where does Sarcoma CellMiner-CDB fit into all of this?
Well, it can tell us the TMB for different sarcoma cell lines.
Okay.
For example, it shows that Ewing sarcoma has a low TMB,
Okay.
which is consistent with what's observed in patients.
Okay. So again, the database is lining up with real-world observations.
Right.
But I'm guessing there's more to this TMB story.
You bet.
Okay.
The database also highlighted two leiomyosarcoma cell lines, SKUT1 and SKUT1,
Okay.
which have the highest TMB among all the cell lines studied.
And what's significant about that?
It turns out that this finding aligns perfectly with a recently discovered subset of leiomyosarcoma that also has a high TMB.
Wow. So the database is helping us identify new subtypes of sarcoma based on their TMB.
Yes.
That's pretty cool.
Absolutely. And it also provides insights into specific gene mutations.
Okay.
For instance, it confirms that about half of chondrosarcoma cell lines have mutations in the IDH genes.
Uh-huh.
And again, this reflects what we see in actual patients with chondrosarcoma.
Okay, so far the Sarcoma CellMinerCDB seems like it's mostly validating things we already know about sarcomas,
Right.
which is great.
Yeah.
But I'm curious,
Sure.
does it offer anything truly new or groundbreaking?
It absolutely does.
Okay, great.
One area where it's breaking new ground is in the study of how cancer cells manipulate their telomeres.
More scientific jargon.
Sorry.
Remind me what telomeres are again?
Okay, so picture the plastic tips at the end of shoelaces.
Okay.
Telomeres are like those protective caps, but for our chromosomes.
Right.
They help prevent our DNA from fraying and getting damaged.
Right, I remember that. But what do telomeres have to do with cancer?
Every time a cell divides, its telomeres get a little bit shorter.
Okay.
Eventually, they get so short that the cell can no longer divide, and it dies. This is a normal part of aging.
Okay.
But cancer cells are sneaky.
Uh,
They've figured out ways to keep their telomeres long so they can keep dividing uncontrollably.
I see. So they've basically found a way to cheat death.
Exactly. One way they do this is through a pathway called ALT,
Okay,
which stands for alternative lengthening of telomeres.
gotcha.
And guess what?
What?
Sarcoma CellMinerCDB can help researchers study this too.
Okay, I'm officially impressed. Tell me more about how the database is shedding light on this ALT pathway.
Well, for example, it helped researchers identify potential ALT-positive cell lines by looking for those that don't express the PERT gene,
Okay,
which is a key player in telomere maintenance.
gotcha.
This discovery could be incredibly useful for developing therapies that specifically target the ALT pathway
Yeah?
and stop cancer cells from basically becoming immortal.
Wow. So we're not just talking about confirming existing knowledge.
Yeah.
This database is actually pointing us towards potential new treatment avenues.
You got it.
That's huge.
It is, and there's another fascinating area where Sarcoma CellMinerCDB is making waves the analysis of the methylome.
Okay, you're going to have to break that one down for me, methylome. I'm drawing a blank.
All right, so think of it like a complex control panel
Okay.
for gene expression.
Uh-huh.
It's a pattern of chemical tags on DNA that can switch genes on or off.
Okay.
And this database is loaded with information about the methylome in sarcoma cells.
So by analyzing these chemical tags, researchers can get a better understanding of how different sarcomas behave.
Exactly. For instance, Sarcoma CellMinerCDB can accurately classify different subtypes of sarcoma
Wow.
based on their methylation profiles. It can even tell the difference between subtypes of rhabdomyosarcoma, which can be incredibly difficult to do.
Wow, so it's like this database is giving us a whole new way to classify and understand these complex cancers.
It is.
That's incredible. But I have to ask, what about treatments? Can this tool actually help us find better ways to treat sarcoma?
That's the million-dollar question,
Yeah.
and the answer is a resounding yes.
Okay, good.
One of the most exciting things about Sarcoma CellMinerCDB is its ability to predict which drugs might work best
Okay.
for different sarcoma subtypes.
Hold on. Are you saying this database could help personalize cancer treatment?
You got it.
Wow.
For instance, it confirms that rhabdomyosarcoma is generally sensitive to dactinomycin
Okay.
and osteosarcoma to methotrexate. These are treatments that doctors are already using,
Uh-huh.
so it's reassuring to see the database aligning with what we see in clinical practice.
That's definitely a good sign. But what about new treatments? Can this tool point us towards therapies that we haven't even thought of yet?
It can indeed. For example, the database suggests that Ewing sarcoma might be highly sensitive to irinotecan.
Okay.
This is a drug that isn't typically used as a first-line treatment for this type of cancer.
Wait, so this could potentially change how we treat Ewing sarcoma?
Potentially, yes.
That's a good deal.
It is, and it gets even more interesting.
Okay.
Sarcoma CellMinerCDB also sheds light on potential biomarkers that could guide treatment decisions.
Okay.
For example, it shows that SLFN11 expression is linked to sensitivity to both irinotecan and the PRP inhibitor talazoparib in Ewing sarcoma.
Okay, break that down for me. What does that actually mean for patients?
Well, it means that SLFN11 could be a key to unlocking more effective treatments for Ewing sarcoma.
Okay.
By testing patients for SLFN11 expression, doctors could potentially tailor treatments to individuals,
Okay.
increasing their chances of success.
This is all so fascinating. It sounds like this database could revolutionize sarcoma research.
It has that potential, but we've covered a lot of ground already.
We have.
Would you like to take a moment to process all this before we dive into some even more mind-blowing aspects of this tool?
You bet. I need a minute to let all this sink in this here. Sarcoma CellMinerCDB is truly remarkable. We'll be right back after a short break to explore even more of its capabilities.
Sounds good.
Okay, I'm back and ready for more. Before the break, we were talking about how Sarcoma CellMinerCDB is uncovering new possibilities for personalized sarcoma treatment.
Yes, we were.
Where do we go from here?
Let's talk about how this database helps us understand
Okay.
why some sarcomas respond well to certain therapies while others don't.
Good.
The researchers actually used it to test the effectiveness of 7 standard sarcoma drugs
Correct.
across all the different cell lines.
Okay, so they're basically using the database to run, like, virtual drug trials.
You could say that.
Okay, cool.
And the results were pretty interesting.
Okay.
The data confirmed what we already know about some drugs. For example, rhabdomyosarcoma tends to respond well to dactinomycin
Okay.
and osteosarcoma to methotrexate.
So it's backing up what doctors are seeing in their patients.
Right.
That's reassuring. But I bet there were some surprises too, right?
There were.
Okay.
The database revealed that Ewing sarcoma might be highly sensitive to irinotecan,
Okay.
a drug that's not usually a first-line treatment for this cancer.
Wow. So this database could actually shift how we approach treatment for certain sarcomas. That's huge.
It's definitely an exciting possibility, and it highlights the power of this tool
Yeah.
to help researchers explore new treatment avenues that might not have been considered before.
So it's not just about finding new drugs, but also about using existing drugs in smarter ways.
Exactly. It's about finding the right drug for the right patient.
Okay.
And that brings us to the topic of targeted therapies.
Targeted therapies, those are treatments that zero in on specific molecules involved in cancer growth.
Precisely. Think of them like guided missiles, designed to attack specific targets on cancer cells
Yeah.
while leaving healthy cells relatively unharmed.
Okay.
And Sarcoma CellMinerCDB can help us identify those targets.
How so?
Well, researchers can use the database to look at the expression of surface markers
Okay.
across different sarcoma cell lines.
Oh.
Surface markers are like flags that sit on the surface of cells.
So these surface markers could act as beacons,
Yes.
helping targeted therapies lock onto the cancer cells.
That's a great way to put it, and the researchers found some really interesting patterns.
Okay.
For instance, they discovered that 2 chondrosarcoma cell lines, SW1353 and JJ01,
Okay.
have high expression of a marker called CD33.
Okay, but isn't CD33 typically associated with leukemia, type of blood cancer?
Right.
What's it doing on sarcoma cells?
That's the interesting part. There are already approved drugs
Okay.
that target CD33 for treating leukemia.
Uh-huh.
This finding suggests that those drugs might also be effective against certain chondrosarcomas.
So it's like repurposing a weapon from one battle to another. That's some serious outside-the-box thinking.
It is, and it's a testament to the power of data integration.
Yeah.
By bringing together information from all these diverse sources,
Uh-huh.
we can start to see connections that might have gone unnoticed before.
There were also 4 surface markers highlighted for Ewing sarcoma, SLC05A1,
Right.
NPY5R, PCDH17, and CDH8. What's the story there?
These genes showed high expression in Ewing sarcoma cell lines, but were relatively low in most normal tissues.
Okay.
This makes them particularly interesting
Yeah.
as potential targets for therapies like antibody-drug conjugates
Oh.
or cellular immunotherapies.
Okay, I'm going to need you to break down those terms. Antibody-drug conjugates.
Okay, think of antibody-drug conjugates like smart bombs.
Okay.
They consist of an antibody which acts like a homing device attached to a powerful chemotherapy drug.
Wow.
The antibody zeroes in on the surface marker on the cancer cell,
Yeah.
delivering the toxic payload directly to its target.
Wow, that's pretty high tech. And what about cellular immunotherapies?
These treatments are all about empowering the body's own immune system to fight cancer.
Okay.
One example is cellular T-cell therapy.
Okay.
It involves taking a patient's T cells, which are a type of white blood cell,
Uh-huh.
and genetically engineering them to recognize and attack cancer cells that express specific surface markers.
So these 4 surface markers they identified in Ewing sarcoma could potentially pave the way for new and more effective treatments.
That's the hope, and Sarcoma CellMiner-CDB is giving researchers the tools to explore these possibilities. It's incredibly exciting.
I can definitely see why you're so enthusiastic about this tool, but I have to ask, are there any limitations? Is there anything it can't do?
That's a great question. One limitation is that the database relies on cell lines,
Right.
which are cancer cells grown in a lab dish.
Right. Cell lines are useful for research, but
Yeah.
they're not quite the same as real tumors in a living organism, right?
Exactly.
Okay.
They lack the complex interactions that happen within the human body. And they don't fully capture the genetic diversity we see in real tumors.
So while Sarcoma CellMiner-CDB is a powerful tool, it's important to remember that it's just one piece of the puzzle.
Absolutely. It's essential to validate the findings from this database in more complex models,
Okay.
like patient-derived xenografts or even clinical trials.
Speaking of which, I noticed the researchers mentioned the possibility of developing a similar tool for patient samples.
Oh.
What would that look like?
Just imagine a database that incorporates not just cell line data,
Right.
but also information from actual patient tumors.
Yeah.
That would provide an even richer and more nuanced understanding of sarcoma.
That would be incredible, but I imagine it would be incredibly challenging to build such a database.
It would be a massive undertaking requiring collaboration between researchers,
Yeah.
clinicians, and institutions all over the world, but the potential benefits are enormous.
It could lead to even more personalized and effective treatments, ultimately improving outcomes for patients with sarcoma. That's a goal worth striving for.
Absolutely, and it would accelerate the discovery of new therapeutic targets and biomarkers,
Yeah.
driving the development of even more innovative therapies.
This deep dive has been so eye-opening. It's clear that Sarcoma CellMiner-CDB is a game changer in the fight against sarcoma.
Hmm.
But before we wrap things up,
Okay,
there's one thing that's been bugging me.
what's that?
The article mentioned that soft tissue sarcomas, including rhabdomyosarcoma,
Yes.
have higher levels of global promoter methylation compared to bone sarcomas.
Yes, that was a fascinating observation.
But what does it mean? Why is that difference in methylation important?
That's a great question, and it warrants further investigation.
Okay.
It could be that this difference in methylation reflects distinct cellular origins or developmental pathways for bone and soft tissue sarcomas.
So it's like a clue that points to fundamental differences in how these cancers develop.
Precisely, and it highlights the importance of studying the epigenome,
Okay.
which includes modifications like methylation in addition to the genome itself.
It's amazing how much we're still learning about these complex cancers. Every new discovery seems to open up a dozen more questions.
That's the beauty of science.
Yeah.
It's an endless pursuit of knowledge,
Yeah.
and tools like Sarcoma CellMiner-CDB are giving us the power to ask and answer increasingly sophisticated questions.
Okay. I'm ready for the final piece of the puzzle. What thought-provoking question did you have for our listeners?
Remember those potential surface markers we talked about for Ewing sarcoma?
Yes.
SLCO5A1, MPY5R, PCDH17, and CDH8?
Yes.
They're highly expressed in Ewing sarcoma.
Mm-hmm.
But low in most normal tissues, could those be promising targets for developing new diagnostic tools or even therapies?
That's a great question to leave our listeners with. It really underscores the potential of this database to not only advance our understanding of sarcoma,
Yeah.
but to translate that knowledge into tangible benefits for patients.
Exactly. Sarcoma CellMiner-CDB is a powerful new weapon in our arsenal against these challenging cancers. It offers hope for improved diagnosis, more effective treatments, and maybe even a cure someday.
This has been an incredible deep dive. I'm walking away with a much deeper appreciation for the complexity of sarcoma research and the incredible progress that's being made.
It's been my pleasure to share this journey with you. Until next time, keep exploring, keep questioning, and never stop learning.
Welcome back. I'm still processing everything we've learned about Sarcoma CellMiner-CDB.
Yeah, it's a lot.
It's mind-blowing how much information is packed into this tool.
It really is a goldmine for researchers.
Yeah.
And remember, it's publicly available.
Oh, that's right.
So anyone with an internet connection can access this treasure trove of data.
Wait, really? So anyone could potentially just dive into this database and start exploring?
Absolutely. That's the beauty of open access science.
Yeah.
It fosters collaboration and allows researchers all over the world to share data and build upon each other's discoveries.
That's amazing. It's like a global community of scientists working together to solve this puzzle of sarcoma.
I like that analogy. And Sarcoma CellMiner-CDB is their high-tech detective kit.
Yeah.
It's going to accelerate progress in this field tremendously.
Okay, before we wrap up, you mentioned having one last thought-provoking question for our listeners.
I do.
What is it?
Well, we've talked a lot about how Sarcoma CellMiner-CDB is confirming what we already know about sarcomas,
Right.
but it's also revealing intriguing new insights.
Okay.
One thing that stood out to me was the potential of those surface markers we discussed.
You mean the ones that were highly expressed in Ewing sarcoma but low in most normal tissues?
Exactly. SLCO5A1, NPY5R, PCDH17, and CDH8.
Right.
They seem like promising targets
Yeah.
for developing new diagnostic tools or even therapies.
Okay, I see where you're going with this. We could potentially use these markers to detect Ewing sarcoma earlier, or even develop targeted therapies that specifically attack cells expressing those markers.
Precisely. It's still early days, of course.
Yeah.
But Sarcoma CellMinerCDB is giving researchers the clues they need to start exploring these possibilities.
So the question you want our listeners to ponder is,
Could these surface markers be the key to unlocking new and more effective ways to diagnose and treat Ewing sarcoma?
that's a great question, and it highlights the potential of this tool to not only deepen our understanding of sarcoma,
Yeah,
but also translate that knowledge into tangible benefits for patients.
exactly. Sarcoma CellMinerCDB is a beacon of hope in the fight against these challenging cancers.
It's inspiring to think about the impact this database could have on the lives of people affected by sarcoma.
It truly is.
Well, on that note of optimism, it's time for us to surface from our deep dive into the world of sarcoma research.
It's been a pleasure exploring this fascinating tool with you.
Thank you so much for guiding us through it, and thanks to all of our listeners for joining us on this journey. Remember, knowledge is power. Keep learning, keep asking questions, and keep diving deep.